Thank you for the introduction. Welcome to our webinar on Toward Global IDMP Implementation, a focus on biologics. Thank you for attending today. We continue to support and encourage the implementation of the IDMP standards. We have made tremendous progress as we work to ensure that the standards can be implemented for global use. TJ and I invited Vada Perkins to participate today because of his extensive international experience and expertise with the IDMP standards for both drugs and biologics. You will hear from him shortly. Today, TJ will present an overview of the ISO standards and the challenges that we have encountered in trying to implement them globally. I will discuss the cross-region and international collaborations that we have established in order to have organizational structures in place to better address the challenges we face with the ISO IDMP standards. Finally, Vada will present on IDMP use cases for drugs and biologics. Now I will toss to TJ.
Thank you, Ron. Let us first look at ISO IDMP standards. The Identification of Medicinal Product IDMP is a collection of 5 ISO standards that are used together to define the data elements and structure that are required to uniquely and unambiguously identify medicinal product, pharmaceutical product, and substance. The standard promote common vocabularies to improve people communication, and it also include common electronic message standard to improve IT system communication. The 5 standards often depict as a diagram you see on the right hand side. The very bottom layer is 11615, which include data element to uniquely identify medicinal product and assign identification to such medicinal product at product level known as Medicinal Product Identification (MPID) and at package level, which is known as Packaged Medicinal Product Identification (PCID). A level up is 11616, which include algorithm to generate pharmaceutical product identification (PHPIDs) based on a combination of substance, strength, and dose form. 11238 define how to uniquely identify substance. 11239 specify how to create and maintain vocabulary for pharmaceutical dose form, unit of presentation, and route administration. 11240 specify unit of measurements to specify the strength of pharmaceutical product. Key benefits of IDMP. With well-defined data elements and common vocabularies, it improves the data quality. With standardized electronic message, it improves the interoperability by making it easier to share information across jurisdiction. With better data quality, easier and fast way to share information, it improves our ability to identify, assess, and respond to patient safety or medication instance faster and more precisely. The standard also make it easier to find alternative product to address product shortage. It also make it easier to compare product in a region or across region. To maximize the benefit of IDMP standards, cross-region or global agreement on common substance ID and dose form is required. Let's take a closer look at PHPIDs. 11616 include algorithm to generate pharmaceutical product ID PHPID based on a combination of substance, strength, and dose form. It further classified the PHPID into 4 levels. Level 1 is generated based on substance only. Level 2 is substance and strength. Level 3, substance and administrable dose form. Level 4, being the most precise level, includes substance, strength, and dose form information. So let's look at how PHPID enable us to connect medicinal product in a region and across region. Here we have a product called Advil liquid gel, which is a medicinal product authorized to be marketed in the United States. The barcode on the right hand side is the NDC code, which is the US implementation of 11615 at package level, also known as PCID. Each capsule is a pharmaceutical product that contains ibuprofen at 200 mg with dose form as capsule. Now, with substance, dose form, and strength, we can calculate level 4 PHPID that would allow us to link to mild or generic ibuprofen by Walgreens or CVS, because all these products share the same substance, dose form, and strength. Now, with cross-region agreement on the term for substance and dose form, we can also link to products like Nurofen in Europe and Ringo in Japan. All these products share the same pharmaceutical composition, so when one of the products causes an adverse event in a region, we can anticipate other products will cause same adverse event in the same region or other regions. And this is how we can do global pharmacovigilance. Also, when there's a product shortage with any one of these products, we can easily find alternative product. And this is how we can combat the product shortage at global level. However, I'd like to get your attention to the dose form for Advil and Nurofen. Advil has a dose form as capsule and Nurofen is soft capsule. US and Europe has different granularity on the dose form. With current IDMP standard, we will not be able to generate identical level for PHPID to connect these two products together. And this poses a challenge to the global implementation of IDMP. Next example is a biologic product. Based on the same principle, we can link products together with the PHPIDs at level 4 PHPID. So we have seen the example with the challenge of different granularity of dose form in different regions. So what are other challenges? When ISO published IDMP standard in 2012, it was based on the assumption that there will be common substance ID and common dose form to generate global PHPID that will allow us to connect product across region. However, as of today, there's no global agreement on common substance ID, hence we are not able to generate global PHPID at any level. There's no global consensus on dose form to generate level 3 and level 4 PHPID. Dose form and strength for the same product can be expressed differently in different regions, which make the generation of PHPID even more difficult. So let's first look at challenge for substance. There is not a set of substance ID that is identified and agreed by the regulatory agencies as global substance ID. When the global substance ID is identified, then a consistent way to assign the identification to a substance will require global maintenance and a global maintenance organization. Then can the global maintenance organization unambiguously identify a substance based on the core set of data field without the confidential information. And then the harmonized capture of standard information is not always well defined for global substance. The issues have been identified by EMA, FDA, and WHO UMC. A working group is established by the parties to work on the potential solution for the global IDMP implementation. Ron will get into the detail on the cross-region collaboration and the working group in detail in the next part of the presentation. I need to mention that the working group recommend that UMC to be established and recognized as IDMP Maintenance Organization, IMO, for substance ID. And also the working group need to identify the process to support UMC as IMO and to conduct pilot to map global substance ID with regional substance ID. Also work with ISO TC215 working group to ensure the framework of capture standardized information, including the minimum data set to uniquely identify substances will support these global substance ID activities. Next is about a challenge for dose form. As we mentioned earlier, there is no cross-region or global agreement on common dose form. Mapping regional dose form is not a viable solution. During 2020 and 2021 ISO TC215 Working Group 6 meeting, the working group accepted a proposal to use a collection of 4 pharmaceutical dose form characteristics as an alternative to generate global PHP ID. A pilot was conducted by FDA and UMC in late 2020, early 2021, and that demonstrates the use of characteristics as an alternative is a viable solution to generate global PHP ID. Based on the finding of the pilot and UNICOM findings, ISO TC215 Workgroup 6 started to revise the ISO 11239 standard and the related technical spec 20440 to include dose form characteristics as a way to generate global PHP ID. The working group also recommends to conduct additional pilot with dose form characteristic mapping with additional regions. Then the challenge with various expression and assignment of dose form and strengths in different regions. First example is about dose form expression with the Pfizer COVID-19 vaccine. EMA approved the dose form as dispersion for injection, FDA is suspension, and UK initially is solution for injection. And then on the strength side, each region may express strengths with different units, such as percentage, international unit, or milligram per milliliter. The example for AstraZeneca COVID vaccine that EMA measure the strength based on infectious unit. Both UK and Australia measure by viral particle, and as you can see, the strength is different and the unit is different. The conversion can be complicated in some cases. The working group further recommends that the regulatory body need to come to agreement on the global substance ID and the global dose form term in order to generate global PHP ID. Also propose to document the PHP ID business rule, including the conversion of strengths and the assignment of dose form characteristics into a new ISO technical report. It also recommends that the WHO UNC be established and recognized as IMO not just for global substance ID, also for global PHP ID. The ISO TC215 Workgroup 6 need to update the standard and provide guidance for the use of different units in different regions. FDA supports and promotes the implementation of ISO IDMP standard and has been working with other regulators and standard bodies to implement IDMP at global level. When the outstanding challenges with ISO IDMP standard are resolved and the process to generate and maintain global substance ID and global PHP ID is established, IDMP will be enabled for global use. With that, I'd like to turn the presentation to Ron, who is chairing the Global IDMP Working Group, also chairing the EMA FDA Collaborating Framework. Ron?
Thanks, TJ. FDA and EMA have been collaborating on IDMP since 2003 in ICH and now through ISO and H07. However, a few years ago, we decided that we needed to establish a formal framework for our IDMP collaboration. So we developed and executed a charter called the EMA FDA IDMP Collaboration Framework. The framework states that our collaboration will be enhanced by way of a governance for IDMP focused on decision-making, authority, and accountability. On a high level, the scope of the collaboration focuses on development, maintenance, and implementation of the ISO IDMP standards. What is important here is that activities to accomplish this need to ensure that the EU and US business, regulatory, and legal requirements are met. The framework has two phases. Phase 1 is focused on cooperation with SDOs, development and maintenance, and GSRS and EU SRS. Phase 2, which we are pretty much in now, is to work on pilot projects and to coordinate with other international activities and activities surrounding global content in support of GSRS and EU SRS. It became clear following the FDA WHO UMC pilot project on dose form characteristics that we needed an organization on a global level that could focus on dealing with the challenges, roadblocks, hurdles, and issues surrounding global IDMP implementation and use. So in 2021, based on recommendations from the 2019 WHO workshop on IDMP, we chartered the Global IDMP Working Group. Currently, the chartered members include EMA, FDA, and WHO Uppsala Monitoring Center. However, the plan will be to include other organizations in Phase 2. Overall, the goal of the GIDWIG is quite simple. Conduct and report on projects that will lead to global implementation of the ISO IDMP standards and maintenance of global identifiers. This is an operational focus group that initiates activities to find solutions to the challenges to global implementation. Overall, and through the recent years, we have observed a convergence in cross-region collaboration on IDMP standards development, their revisions, and now with implementation activities. This slide just shows the separate activities and projects in the EU and FDA and how the two agencies, along with WHO UMC, are now collaborating more than ever through the Global IDMP Working Group. Next topic is solutions for global IDMP implementation. As I noted, the Global IDMP Working Group is all about conducting projects leading to global implementation. TJ presented the challenges, and briefly I will present the pilot projects for 2022 and 2023. Currently, there is no global substance identifier that has been recognized by the regulatory agencies. Global identification of a substance is essential for the generation of a global PHPID. The first pilot project planned is focused on substance. It will leverage all the work that has been done in the EU Substance Registration System projects, including the Vaccines Initiative. The scope would be to map the EU's EU TCT, the US Unicode, and possibly a code from another region to a global identifier for a set of chemicals in the WHO model list. Also, it will be to review more complex situations related to biologics. There has been no recognized single controlled global dose form vocabulary. So in 2021, as part of a work stream of ISO TC215 Workgroup 6, FDA and WHO UMC demonstrated that the EDQM dose form characteristics can be used to describe a marketed product's dose form and could be used as input into the generation of a global PHPID. This second pilot on dose form will look to ensure that there is consistent mapping to the EDQM dose form characteristics for products with less granular dose form expressions. The pilot will map dose forms to another region's vocabulary using the approach developed in the TC 215 Workgroup 6 pilot. The pilot will also investigate dose form combinations and EDQM dose form characteristics with multiple values. The success criteria here would be to identify and address issues and challenges and documented rules for proper dose form characteristic mapping and generation of PHPID. We want to build upon the work done in the FDA WHO UMC pilot on dose form characteristics, where concepts were developed for the use of strength presentations versus strength concentration for different products. There are different patterns possible, such as percentage, international units, milligram per gram, milligram per milliliter, and so on. For example, for tablets, creams, sprays, multi-dose vials. And of course, we will work with ISO on this for further clarification. Pilot 4 will focus on H07 Fire Messages and our participation in the development, verification, and ballot of the fire resources related to IDMP. It is important to be able to successfully exchange medicinal product and substance information between EU, SRS, FDA, GSRS, and the UMC SRS using a fire message. The fifth pilot is to evaluate how the implementation and use of the standards globally would work in a production state. We have just started to design the operating model for global PHPID generation, including the use of a global substance identifier. The scope is to demonstrate a consensus-based model for WHO UMC as the international organization for PHPID validation and maintenance. Here's a high-level diagram of the process, which of course will be more detailed in production. This is a summary roadmap of our IDMP activities since the EMA-FDA collaboration framework was put into place, and our planned activities this year. Some of them will likely cross over into 2023 as well. But this gives you a quick snapshot of 2019 through 2022. Now I would like to pass the baton to Vada, who will present on IDMP use cases for drugs and biologics.
A very special thanks to my FDA colleagues for the opportunity to present to you today on IDMP points to consider for biologics. My name is Vada Perkins, and I'm Executive Director for Regulatory Policy and Innovation at Bayer Pharmaceuticals. As illustrated by Ron and TJ, there are some distinct challenges with being able to identify a medicinal product uniquely on a global scale. Right. These challenges actually exist at regional levels, but so then basically multiply that or compound that with other requirements from other regions in order to truly identify a medicinal product uniquely on an international scale. However many regions you have to accommodate, however many criteria or how many use cases you have, fundamentally it all starts with the ability to scientifically and objectively identify a substance. Right? If we don't get the substance right, then pharmaceutical product identification and medicinal product identification will not be possible. Right? So there are objective defining elements that have been identified in the ISO standards, taking into account expertise from stakeholders around the world to not only associate, but to differentiate substances utilized in medicinal products. Some of those additional challenges that were identified is that we have to be able to determine, what a common substance ID looks like, right? Can we get global agreement and adoption of the standard, understanding there are regional requirements in order to achieve this? And in addition, other challenges are what is going to be the global substance ID and who's going to maintain that over time, right? So when we think about small molecules and we think about molecular structures, that becomes a little bit easier to accommodate, and there's a lot of work that's already ongoing between INN, USIN, and other groups. But when we get into biologics, this is where it gets much more complicated, and we'll provide some examples of the complexity of biologics and what we should be considering in order to achieve unique identification of medicinal products for biologics as well. So just a bit of background on the differences between small and large molecules and those characteristics. These are the things that really do make IDMP, when it comes to biologics, a lot more complex. When we're dealing with small molecules, chemicals, simple chemicals, they're well defined and they're independent of the manufacturing process, right? They're produced by chemical synthesis, identical copies can be made, and the chemical process is fairly predictable. When we're dealing with biologics, much more complex, heterogeneous, defined by manufacturing processes, and they're very difficult to control from the starting material to the final active pharmaceutical ingredient, right? It's impossible to ensure identical copies, and characterization is a lot more complicated. They're bigger and they're organic, which basically lends them to be a bit unstable and sensitive to external conditions, which is much different than small molecules as mentioned. So to further illustrate the point about small molecule versus large molecule characteristics, we have to really look at the size and the complexity of them. And as you can see from this illustration, a small molecule drug such as aspirin is 21 atoms. Very easy to look at that, say the molecular structure in terms of its definition. As you get into large molecule drugs, you'll see that human growth hormone, it's about 3,000 atoms. And a large biologic like Herceptin is 25,000 atoms. One analogy that's very popular in terms of this is looking at vehicles. And you can say that a small molecule or aspirin would be the equivalent of a bicycle, whereas a large molecule would be the equivalent of a car or a truck, and a large biologic would be the equivalent of a military fighter jet. So you can only imagine that the bigger it gets, the more complex things become. And again, this is sort of what we're dealing with in terms of really trying to then uniquely identify a medicinal product. When we get into biologics, that's a level of complexity that's much more different than a simple chemical structure and a molecular formula to define something uniquely. So another example, another way to visualize the complexity of biologics versus chemicals is to look at the manufacturing process. And as I've mentioned, the manufacturing process for biologics is key. It's much different than how you would go about identifying a chemical, which, as we've said, can be defined by its molecular structure. So in looking at tablet manufacturing, not to get into a lot of detail here, but this is simply to illustrate, what you see here is a little bit more straightforward than you'd see with manufacturing biologics. You have your API and your excipient feeders. It goes through a process, and as you can see on the right, when you have your tablet, you can really kind of break it down into different parts of the active ingredient, the disintegrant, a binder, lubricant, and diluent. So not to say it's not complicated, but it's pretty straightforward in terms of how you visualize tablet manufacturing. So biologics manufacturing, in comparison to tablet manufacturing, you can already see from the illustration here that there's a level of complexity with criteria that you must take into account that is simply not the case with chemicals. In dealing with expression systems, master cell banks, working cell banks, culture fermentation and purification for drug substance production, sterile filtration and aseptic filling for drug product production, and a host of ICH guidelines given this level of complexity to assist manufacturers in truly understanding what those regulatory requirements are when it comes to biologics manufacturing. Right. To further illustrate this, on the right-hand side, we looked at the tablet manufacturing process, which is relatively straightforward in comparison. As you can see, cell and raw materials, small and large-scale cell cultures, and then having to recover the product, filter it, formulate it, and then following through that cascade of biologics manufacturing, dealing with sterilization, lyophilization is applicable, and then getting into the packaging of that product. And it doesn't end there. Now we're talking about storage conditions and stability, which is much different to take into account. Right? We're dealing with something that, by comparison, is unstable, given that it's organic, in comparison to a chemical, which is very, very stable. So again, just to illustrate this, given that, as stated, the manufacturing process is critical, plays a large role in how you identify a biologic. So to do a bit of a deeper dive on this process, traditional drug products that we've talked about usually consist of pure chemical substances that are easily analyzed after manufacture, right? But there is a limited ability to be able to do that, to identify the identity of clinically active components of a complex biological product. And again, to reemphasize, because it's very important, that biologics are defined in many ways by their manufacturing process. You could have the same actives for the same indication being manufactured by different companies who may have different manufacturing processes, and those must be taken into consideration, right? Because any changes in the manufacturing process, equipment, or facilities could result in changes to a biological product. There are submissions called comparability protocols, where you're looking at changes that you may have taken in the biologics manufacturing process, and now you're looking and saying, well, are there any differences between the actual biological product as approved in comparison to what we're getting now, given some changes that we've made in the manufacturing process, equipment, facilities, etc. And depending on what those differences are, it could require additional clinical studies to demonstrate a product's safety, identity, purity, and potency. And these are definitely things that occur, and you don't see this when it comes to simple chemicals, right? So how do we get a handle on identifying substances from small molecules all the way through large molecules by the ISO IDMP standards? And the ISO IDMP standard for substances groups these in 5 different categories with associated defining elements for harmonization. Chemicals, which are defined primarily by the molecular structure. Proteins, which can be identified by the amino acid sequence, type of glycosylation. Looking at nucleic acids, also looking at sequence, other modifications. Polymers, whether they be synthetic or biopolymers, and this could be identified in terms of defining elements by molecular weight or properties related to molecular weight, or the biological source. And for structurally diverse substances is where we're talking about vaccines, and we'll go through an example of that. Viruses, cells, tissues, complex materials. There's taxonomic, anatomical elements, other physical properties that really play into that. So really, this is just to illustrate that you go from something very simple, such as a chemical, right? And there's extra credit for those who might identify what that chemical structure is, and just kind of working your way through proteins, nucleic acids, polymers, and structurally diverse, getting into much more complicated molecules, where you have to start using other defining criteria to identify something uniquely. So getting into considerations for unique substance identification versus product identification when we're dealing with naming conventions, and we're dealing with naming conventions with USAN and INM primarily. we are conditioned to look at names. If you've had a chance to look at the FDA's substance registration system or the global substance registration system, or even systems within our own organizations, you'll know that there are many, many synonyms for the same thing, which makes it very difficult to identify something, well, nearly impossible to identify something uniquely, given that there are many names for the same thing. Right? And one example is acetaminophen and paracetamol, even though we're talking about the same structure. With amoxicillin, as an example, you have the same nomenclature, same name at the INN or the USAN level, but for the purposes of unique identification for what we want to identify with IDMP, we have trihydrate and anhydrous, which are different structurally. And if you look at entry terms in MESH, for instance, you can see that there are many, many names that we have to contend with there. And then lastly, I'll use an example of amlodipine, where at the same time, the level of detail or that we need to uniquely identify something when it comes to IDMP goes a little bit further, right? Because we don't want to take something that's structurally different and say it's the same thing at the IDMP level, whereas in many other instances that might be acceptable for non-generic naming, but for the purposes of scientific identification, it requires another layer in order to uniquely identify a substance. So further considerations for unique substance identification versus product identification. That level of depth and breadth that we talked about is extremely important. And with biologics, as mentioned, the manufacturing process is key to how you're going to define that biologic. The examples I gave about USAN and INN, those exist. That is something that is still an ongoing challenge when it comes to biologics with those naming conventions. And given that we're short on time here, I would recommend that people have a look for themselves. You can look at this publication that talks about INN global nomenclature of biological medicines and the continuous challenges that exist with that. And those challenges have a lot to do with what we just talked about in the last few slides, that level of complexity for biologics versus simple chemicals, and how do you address those complexities. So I thought it would be helpful and interesting to go over a couple of examples for unique identification of medicinal products with respect to biologics, starting with vaccines. Right now, what's top of mind and in the news, you can't get away from it, are vaccines for COVID-19. And when we look at those vaccines, there are different manufacturing processes for that. So viral vector vaccines, protein-based, and genetic vaccines. So these are the genes administered directly as either DNA or RNA. And we're more familiar now with mRNA, given what's been in the news because of the Pfizer and the Moderna vaccines, which are formulated utilizing this technology. Again, the illustration here is not to go into details, but just to further take home the message that when you're looking at simple chemical structures, that biologics are much more complex, as you can see here. Another example happens to be insulin and influenza. Right? Insulin's interesting because in February 20th of 2020, the FDA published the final rule that updated the term biological product. So prior to that, insulin wasn't in that category. Right? So this tells you the evolution of thinking about how we need to get into more scientific identification of medicinal products. And in this final rule, a biological product now includes all proteins, which are defined as an alpha amino acid polymer with a specific defined sequence that's greater than 40 amino acids in size. So interesting that, like I said, in 2020, insulin is now a part of that biological product definition. And then when you're dealing with influenza, which is possibly also top of mind for some, given the season that we're in, that depending on the process, even though you're dealing with the same strains, depending if something is egg-based or cell-based, depending on the inactivation method, These are defining elements and criteria that you'd say, okay, yes, we're dealing with the same strain, we're dealing with the same indication. However, in the manufacturing process, there are some differences which would then say, well, these are then different substances. So getting into some more detail about the coronavirus vaccines, I mentioned that this is just an example that to further illustrate this manufacturing process, right, where these differences, even though you're dealing with, say, it's the same variant and we're doing it for the same thing. You can see here, nice illustration from NIH, that for a protein-based vaccine, the spike protein is purified and injected. With a viral vector vaccine, the spike protein gene is purified, adenoviral vector is injected. Your body produces the spike protein, and then the immune system produces the antibody. And then lastly, with mRNA, such as with Moderna and Pfizer, the mRNA that codes for the spike protein is purified and injected, and that the body then produces a spike protein, and the immune system produces the antibody. So to provide a little bit more detail in terms of the complexity of biologics, let's just take the mRNA vaccines as an example for unique identification, right? So not talking about viral vectors or the protein-based, but just the mRNA vaccines. And as you can see here, and I pull this from the FDA's substance registration system, you can see that for the Moderna COVID-19 vaccine, you can see what the active is here. It has its preferred substance name that corresponds to what is in the INN and the WHO Drug Dictionary. You can see other synonyms here, but the most important thing is that you look at the name, you look at the uni, that this has a very specific substance associated with this. Then getting into the Pfizer COVID-19 vaccine has a different active, right, with a different uni and a different preferred substance name. So even though we're dealing with the same technology, mRNA technology, experts in this space certainly have looked at this and said, you know what, even though you're dealing with COVID-19 and this variant and mRNA technology, the way these are manufactured, these are different substances, right? And again, for those who are very much experts in this space, they can articulate this a lot better than I can, but what I offer you is a publication that gets into some of the details here, and as you can see from one of the criteria from the screenshot, that there are differences in formulations. So if you look at this illustration about the active ingredient and the company, you can see here, just as a method of illustration, how there are some differences that must take into consideration. So again, even though we're talking about COVID-19 and we're talking about the same technology in terms of how the vaccine is manufactured, many other considerations to take into account for biologics in this space. So what now? Well, there, as Ron and TJ have illustrated and have mentioned in their presentation, there are a lot of experts in a lot of regions that are working collectively to basically try to solve this issue, right? And it's work that's been ongoing for years. It'll probably continue to take many more years to really see true global harmonization. But what can we all do, right? If you're a regulator, you're probably working in this space, have colleagues in this space, but for those of us in industry or we're vendors or we have just a genuine interest, it's really about observation and engagement. So if you look at what's been presented, there's already an EMA FDA collaboration framework charter that Ron presented. So it's really important to see that, okay, there's proactive engagement on this topic for global identification. Let's really try to observe and see what comes out of that and see where there are opportunities to contribute to that. You have the EU SPORE initiative for IDMP implementation in Europe. The US FDA roadmap, something to observe and keep an eye on to really get the feedback from the agency on what they're doing for 2022 and beyond. And for me personally, I think the global IDMP working group that's been established really is to me paramount in terms of having some type of governance structure to look at how this can be done globally. And so it's important that regions are doing their work, but even more important that regions that are engaged in IDMP implementation come together globally. And it shouldn't just be health authorities, because this really takes a lot of expertise. It really does take industry engagement, scientific experts from other domains in order to contribute to this. And you can see here that there's allocation for observers. I had the opportunity to participate in one of these meetings as an observer from industry, and I think it's an important activity to keep an eye on. And as that sort of evolves and develops, it'd be interesting to see where there could be further opportunity for engagement. Trade associations. So if you're in the industry and your company belongs to one of these trade associations, both trade associations listed here, actually all three, have a lot of IDMP activity ongoing. And then there are IDMP specific forums. You can look at the IRIS forum, which is a not-for-profit that has many, many topic groups. One topic group in particular, aside from CMC or digital regulatory or EPI, is IDMP. And that group has been established and been in place for quite some time, very active, with opportunities to hear from health authorities and other experts about what's happening in the IDMP space globally. And then there's also regional activities such as UNICOM, which is a EU-commissioned funded initiative for the implementation of ISO IDMP in Europe. This is something that goes beyond the regulatory domain into the clinical domain, right, in the healthcare domain, dealing with cross-border prescriptions, counterfeiting, drug shortages, etc. And then I'll leave you with an infographic that was created from Unicom that you might find interesting. It's called IDMP in a Capsule. So for those of you who are looking for, I'd say another tool to kind of look at what the benefit of IDMP can be beyond the regulatory environment and how it really should work once we achieve global harmonization in the healthcare domain and that clinical domain, I would encourage all of you to look at this infographic developed by a colleague of ours from ISO, along with other colleagues in Unicom, and it truly is a very nice infographic and really does present IDMP in a capsule. So it's a very important resource to understanding and tracking the requirements for unique identification of biologics and chemicals, those types of substances as well. I would say almost go no further than one, having the standard available, the ISO 11238 standard, which goes into a lot of detail, and Dr. Lawrence Callahan and Dr. Frayne Switzer from FDA who are co-authors of that standard, but the work that they're spearheading along with other colleagues at FDA, NCATS, and even with European health authorities, because there's a collaboration transatlantically here as well, is for this global substance registration resource. It's open source. Industry, vendors, interested parties can participate. They have this global ingredient archival system notification list. You can be informed about what types of changes and updates they're making, but it's going to be virtually impossible to try to remember off the top of your head or through any notes to say, well, what are the defining elements for a protein, for nucleic acid, for structurally diverse substances, vaccines, and you get into the cell and gene space. Very, very difficult. Fortunately, this substance registration system is following the ISO standards with respect to the defining elements. You'll be able to keep track of things there and use it as, I would say, as a resource, but also as a framework to how you're really going to deal with structuring or managing substances in your own internal environments in your companies. Right. So again, very, very important resource that I certainly would encourage everyone to utilize to kind of keep track of what's happening with substances and more importantly, the complexity of biologics. Thank goodness someone's keeping track of this in a lot of detail to make sure that it's compliant with ISO and new scientific advancements so that we can kind of stay clear on how to do this from a global harmonization perspective. So in summary, there are distinct points to consider for identifying biologics versus chemical substances. There are different considerations for different types of biologics. And a global PHPID network solution is absolutely not feasible, will not work if substances are not uniquely identified on an international scale, right? And that's going to require a global maintenance organization. So once we've basically sort of cracked the nut of all the rules, who's going to maintain this for substances and pharmaceutical products? And that's key. And it's fortunate that WHO Uppsala Monitoring Center is looking to step into that space and contribute their resources and expertise to do that, because that is definitely what's going to be necessary once we've solved all of the challenging issues, to have someone who can operationalize that and maintain that globally over time. And again, the manufacturing process is critical to global and unique identification when it comes to biologics. I cannot emphasize that enough. It would be nice if it was basically we're able to identify them quite simply using a simple molecular structure. But as I've indicated, that is certainly not the case. And depending on the level of complexity for the biologic that you're dealing with, you got a lot of criteria that you have to take into consideration, right? So nomenclature and robust scientific identification is foundational. It must occur. The process by which small molecules are identified is just not sufficient for a large molecule identification. So I'd almost look at it and say there's INN and USAN for the purposes that we use it for. But just like ESPN or Disney Plus, when you're adding a little bit of additional service or scope, we should be looking at what's occurring in that space in addition to what is occurring in other spaces to address how we can uniquely identify biologics. So I'd like to thank you for your time and attention today and leave you with the following quote, that the significant problems we have cannot be solved at the same level of thinking with which we created them. I find this to be extremely true with what we're trying to do for global harmonization for the unique identification of substances, pharmaceutical products, medicinal products, for all of the use cases to support public health. And again, it's not just a level of thinking from one region or one organization. It's been very clearly articulated by our FDA colleagues about the level of engagement happening globally, where we as an industry are engaging and when we're dealing with our own scientific and technological advances in terms of new modalities, novel products, and of course dealing with the vendor community and other scientific experts on how we can really achieve this collectively. And lastly, I would like to truly acknowledge and thank FDA colleagues and friends listed here for their input over the years and really shaping this topic and making it what it is today in terms of the level of international awareness and engagement, the scientific expertise, the subject matter expertise and resources that have been dedicated to this topic. Certainly still a long road ahead, but a personal thank you to Ron and TJ for providing this offering on this topic. And then special thanks to Larry Callahan, Frank Switzer for their continued leadership and expertise in the substance space, and then continued leadership strategically from additional FDA colleagues, Joan Blair and Mary Ann Slack. I'd like to thank everyone again for their time and attention, and to turn it back over to you, Ron and TJ, for the Q&A. Thank you.
On behalf of Vada and TJ, I want to thank you for attending today. We hope this webinar helped you to better understand IDMP, its implementation challenges, solutions, and use cases. We will continue our work and collaborations and report back in the future. Thank you.
Thank you for the great presentations. We'll now transition into our Q&A panel session. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Looks like we have some questions coming in right now. And the first group of questions will be directed to Mr. TJ Chen. And here's the first question. How far has the recommendation about who UMC's new role gone, and where could we get updates?
Thank you. I hope you can hear me well. That is, in fact, is a very good question. So the EMA, FDA, WHO, UMC initiative, the working group met a couple of times and then drafted that 5 projects presented during the WHO headquarters held stakeholder meeting. The role for the WHO UMC to be the global substance ID and PHPID maintenance organization was proposed and endorsed by the WHO headquarters, also EMA, FDA senior management. Now, with that said, as you see, the 5 pilots, especially the last one that Ron mentioned, is about a process. The pilot tried to figure out how to make WHO UMC to become the IMO and with effective, efficient way to maintain the IDs. Also need to find out a process that can make the UMC sustainable. So this is, of course, with a lot of commitment, but the pilot will figure out more. So I hope that answers your question. Thank you.
Thank you for responding to that question. We've got another question from Mr. Chen, and here's the next question. Do you have more material on the pilot conducted by the ISO work group and UMC?
Again, thank you. Thank you for the question. So WHO, well, actually ISO TC215 published some information. And I may also miss a second part of the previous question. I think these 2 questions kind of related to information being available. WHO headquarters is setting up a web page that we may post some information since the work group just formed, and the project, the detail and the scope of the project is to be discussed in February. So once we get all the detail and that, we may start to populate that web page. And also EMA and FDA may also create some linkage to that web page. So information will be updated when we get to more detail. Thank you.
Thank you for responding to that question. We do have another question coming in, just arrived for Mr. Chen, and here's the question. Will FDA post ISO documents on their website, and will the posted ISO documents be available for no cost?
All right, thank you. Now, FDA does not normally post document by the standard development organization like CDER, H07, and especially the ISO document are proprietary and need to be purchased. So we are not able to post those information for free access. Thank you.
Thank you for responding to that question. The next group of questions will be directed to Dr. Fitzmartin. And here's the first question. There is almost no information about GIDWG and its activities online. Where could we get more info and updates?
Yes, thank you, Ray, for that question. Well, the reason for that is the global IDMP working group is relatively new. We stood it up just late last year. and chartered it. And we are, in the past several months, looking to stand up a web page. And WHO has offered space for us to have a web page for the Global IDMP Working Group. So stay tuned. We will have a presence, a public presence in the near future. Until such time, we will be doing more webinars such as this, but we will have that website up soon enough. Thank you.
Thank you for responding to that question. The next question for Dr. Fitzmartin, we just had roll in, and here's the question. Is it expected to include industry representatives for like pharma and EFPIA and the global IDMP working group like the IDMP SPOR task force in Europe? And if yes, when and how would we register for that?
Yes, thank you for that question. At this point, the chartered members of the GIDWIG are EMA, WHO, UMC, and FDA. However, the charter does state that once we get into a phase 2 of the GIDWIG, and what does that mean? Once we get to the point where we have the pilots, the 5 pilots that you heard about this morning underway, it is likely that we will want and need participation by not only other regulatory agencies, but also participation and involvement by industry as well. Thank you.
Thank you for responding to that question. We have one more question for Dr. Fitzmartin. And here's the next question. Does FDA have a view on the process of how to exchange IDMP information for like substances or PHPID registration and for products from industry to authorities such as the FDA, UMC? And when would these exchanges of information start to occur?
Thank you. Well, this is almost like a multi-part question and answer. So there's IDMP standards. Some of the information that is exchanged now regionally conforms with the IDMP standards. For example, the NDC code conforms with the NPID. The Unicode for substances conforms with the ISO 11238 standard. So that's happening now and is clearly specified in some of the tech specs for ICSRs, for example. As far as PHPIDs, we're certainly not there yet, especially on as you've heard, on a global scale. We're not there as far as being able to generate a global PHPID because there's 2 missing pieces. The global substance identifier that we can agree on, and a solution for dosage form. And we, as you heard from TJ, we do have a solution, that being the dose form characteristics. And a lot of that work is being handled through the revision of the ISO documents in work group 6 of TC215. And so As far as exchange of information, you also heard that one of the work streams will be a collaboration on H07 Fire Message for the exchange of IDMP medicinal product information as well as substance information. So a lot of this work has to be done before we can get into, from what we would say of FDA, actual use of these IDMP standards for their global intent, which would be to generate a global PHPID. But we are moving in the right direction. As you heard today, there's a lot of work that needs to be done, but it's very exciting that this work, in fact, is being done. Thank you.
Thank you for responding to that group of questions. We've got some more questions coming in right now, and the next group of questions will be directed to Mr. Perkins. And here is the first question. Will there be any immediate impact on the content of Module 3 sections for drug substances of BLA?
No, thank you for the question. So I would say the short answer is no. What we're really dealing with IDMP isn't to impose new regulatory requirements where health authorities aren't doing that. It's really just figuring out from what's already required in Module 3, how do you represent that information to support unique identification. So I don't know if Ron or TJ want to add to this, but the intent, because there's no enforcement, it's not about adding new requirements now. It's just about how do you deal with the information that we're already gathering and make sense of that to support global identification.
Thank you for responding to that question. We've got another question for Mr. Perkins that just came in, and here's the question. Given all the nuances with how biologics can be identified, does the IDMP standard go far enough to allow for meaningful processes to be utilized with the data, for example, safety processes?
So that's an excellent question. And I would say is that everything's a work in progress. I'd say that in the immediate, for the immediate answer in terms of just defining things uniquely at the substance level, that information exists in the standards. Much of what you see in the Global Substance Registration System, those defining elements for the types of substances that exist. When you start getting into the details of the manufacturing process, what we call the specified substance in that group 4 specified substance, that requires a lot more work. So I'd say in principle, what is needed is there, but in terms of what is being captured and what's been identified to leverage that, to do that, that doesn't exist yet. But fortunately, like I said, the groundwork or the foundational pieces to identify a substance at that level uniquely exist. And then there's just more work that needs to be done over time to get to the level of detailed to identify or represent what you're talking about.
Thank you for responding to that question. We have one more question for Mr. Perkins in this round. And here's the question. Does IDMP apply to cancer vaccines? And could you describe this, please?
Yes. So the easiest way to determine, if it's in scope is, what's regulated today. And if you look at, if it falls in one of the categories of the substances that we've talked about, so whether it's chemicals, proteins, polymers, structurally diverse substances, and vaccines are specific to that, and there's an annex specific to vaccines in the ISO standards, the answer is yes. So whether you're talking about, depends on how you define it, even the HPV vaccines or the, which are more therapeutic, but then like say Sipuleucel or therapeutic, in that therapeutic area, the cancer vaccines, how you define them, if the substance is captured within the scope, which it should be, then those products are captured as well. Now, it could be more details and more criteria that have and work that needs to be done for that specific case, but in terms of its scope, it is captured within the IDMP standard. That's the intent.
Thank you for responding to that question. The next question is a special question. It appears that it'll be addressed to Mr. Perkins first, and then he'll be able to pass over once he's finished to TJ for any follow-up comments. And here's the question, and the first is a comment. I'm surprised to hear the speaker state there's no global substance identifier. Isn't this what the INN name does, except for some products like therapeutic vaccines, or excluded from INN? And what is the difference between INN or USAN or other national equivalents versus the global substance ID that is envisioned?
Yeah, so to start, I think from the examples that we gave, INN doesn't have that level of granularity that's been identified in the IDMP standards for substances. When we get into some of those details, as we mentioned with biologics, there's a lot of defining elements. Even when you look at chemicals, right, when you're looking at that still has a granularity piece where INN, it's sufficient for their purposes, but then for unique identification at the substance level, that's where there's more. And it's not that it's not associated. If you saw from the examples, the INN names are associated with what's been identified, for instance, with a uni. There's CAS numbers, there's other things that are out there. But that's the reason why you say there's no global substance identifier. And even in the question, I think it talks about products and substances, and I think that's where there's some confusion, right? Products can be defined, and then substances have to be defined, and the two aren't necessarily, they're not lined up one-to-one, right? So either you're identifying products, you're identifying substances, but a substance identifier in terms of IDMP is not a product identifier.
Yeah, right. So, like Veda mentioned, right? INN is normally is at moiety level. It often does not go down to the salt or hydration as identified or specified by the ISO IDMP standard. However, with that said, INN is still, when we do this global substance ID project, INN is going to be treated as a seed for us to go down to each of the substance. INN, actually, the WHO INN group has participated in the Global ID Working Group meeting, provide very good input, and we're looking to have them also join the Global Substance ID project. Thank you.
Thank you for responding to that group panel discussion, and then we'll go back. The next group of questions will be directed to Mr. Chen, and here's the next question. Can you comment on the progress towards a global substance IDs? And could you also comment on the progress towards establishing a global ID for manufacturing sites?
Thank you. I would take the substance ID. The manufacturer ID side is not, it is for now, it's not in the scope. The global substance ID, the idea is to have UMC to stand up a system that would map FDA, like Ron mentioned that earlier. It will map FDA's UNI and EU's, I think it's EU TCT, to the global substance ID. And the idea is to generate this ID that is neutral, it's not depend on each region. However, they will be mapped and linked together. And that particular ID, the global substance ID, will be used to generate a PHP ID. So that is the idea. The project is to be start meeting in February. The detail on the scope will be selected, and then the progress, the milestone and all that will be then determined during that meeting. Thank you.
Thank you for responding to that question. Looks like we've got a couple more questions from Mr. Chen, and here's the next question. Is it the case that the pilots presented will be completed all in 2022? And if not, what is the target completion timeline for these pilots?
That's a good question. That kind of related to my answer earlier. So the pilot, again, the scope for each of the project will be defined by the project team. However, at this moment, we have a template kind of layout already. We're looking for a more agile approach. So at this moment, a high-level guidance to each of the project team is to produce a 3, 6, 9 month milestone. So, and then depends on the scope, right? Because some may take, I mean, the substance example, I mean, if they take a small set, it will be done in 3 months. If they take bigger set and even include biologic, that is going to take much longer, right? So we have this agile approach for the project lead to discuss with the project team, so they can create this 3, 6, 9 months milestone and report to the greater community. So I hope that answers your question. So the idea is to do most of the thing in 2022, like Ron mentioned in his presentation. Some may linger into next year. For example, the fire project, it need to go with H07 vetting process. So that may not be totally controlled by the project team, and it can drag on longer than we expected. Thank you.
Thank you for responding to that question. We've got one more question for Mr. Chen, and here is the question. What organization will agree on or deliver the global substance ID?
Right, so that, like Ron has presented earlier, we are now looking for WHO UMC to be the organization to maintain that substance ID. To do this activity, we need an organization that has the experience and the prospect at global scale, right? I mean, all the regulators are very good at regional thing. We can do good in what we do, but when it comes to global, we need someone that has done it before and is willing and capable to do it. So now we're looking to WHO UMC to take that role and to generate and maintain substance ID and PHP ID. Thank you.
Thank you for responding to that group of questions. The next group of questions we'll go over, we're bringing back in Dr. Fitzmartin, and here's the first question. Can you comment on any FDA guidances for IDMP?
Thank you, Ray. Any specific guidance for FDA? Currently, we don't have a guidance on IDMP standards. We certainly, as we mentioned today, certainly support and promote the implementation of the IDMP standards. However, as we stated earlier, the IDMP standards are noted in a number of documents and standards that are currently in use. For example, the E2B ICSR standard references the IDMP standards clearly in its documents. So the standards are being used. Certainly, there's placeholders for PHPID in the specifications, but we're not there yet, as we mentioned. But eventually we will be. So as far as a guidance, FDA doesn't have a statute, doesn't have a regulation, and currently doesn't have guidance on IDMP standards, unlike Europe, where they do have a regulatory mandate. But as you heard today, we certainly are supporting the implementation and eventual use of the standards globally. Thank you.
Thank you for responding to that question. We have the next question for Dr. Fitzmartin, and here it is. Is the feedback from the GIDWG work fed back to national agencies?
Thank you. Yeah, as I said earlier, the GIDWG is very new. And so we don't even have our web presence yet. But the plan will be to be as transparent as we can to provide information on the working group. And so soon you will see a lot of the work that is underway posted to the WHO webpage. And so you'll see all of the information about the pilots. As TJ just mentioned, we're looking for an agile approach to the pilots, 3, 6 month, 9 month milestones. And so as developments occur, we want to post that information to the webpage. Thank you.
Thank you, Dr. Fitzmartin. We lost a little bit on the last part of your answer to that question, so I just want to circle back around to you and give you a chance to just summarize what you just said, just to make sure the audience hears everything.
Yeah, sorry about that. Yeah, so we, once we have a web presence on the WHO system that is now being worked on, we will post as much information as we can, presentations and results. As TJ said, there will be an agile approach, 3, 6, 9 month milestone approach. We'll post results to that site as soon as we can. But we're not ready to stand that up yet. We're just got authorization to have a presence on the WHO site. Thank you.
Thank you for summarizing and coming back on and just clarifying that, so we heard you a lot better that time. And we're going to move on to Mr. Perkins it looks like we've got just a little over a minute left for one last question. And here's the question. Once the global PHID is identified, how long do you think it will take to be implemented worldwide in manufacturing environments and educational curriculums?
Yeah, so unfortunately, the answer is it depends. For me personally, my personal take, I think once the global PHPID has been identified, then I think its uptick should be quick relative to all the other work. The challenge, where things are really going to take time, as we said, was the substances, having the substance information, and then what Ron and TJ talked about, having unique dose forms, really getting some agreement on strength. There's so many pieces, precursors that have to be addressed in order for the global PHPID to be identified, that's really what's going to take the time, because as they mentioned, the PHPID is basically an algorithm, right? You're looking at what uniquely you've identified for substances, how you've identified dose forms. If everyone can agree to the same terminology set globally, and we're deploying it the same, then the global PHPID can then basically take care of itself, and then you can focus on, okay, how can you implement this for, say, Unicom and cross-border prescriptions and other things. So yeah, I'd say that's sort of the answer, is that the front end work is what's going to take the longest amount of time, and until we actually get a handle on that, we won't necessarily know when we'll have a global PHPID.
Thank you for responding to that question. We have another question for Mr. Perkins, and here is the question. How has the EU addressed the issue of substance identification as part of the XEV MPD, and can that approach be used globally?
So with respect to where the angle is from industry, I think there's already been a lot of work that's been identified by the health authorities where they're collaborating on the global substance registration system. So within Europe, there's an EU SRS activity. There is a US SRS activity. Ron's already mentioned there's this global formal collaboration between both those organizations. And I do know that the EU SRS team and the US SRS team have been working together. So I would point to that. I think there is a global ingredient archival system group that's open to industry to keep track of those activities. And it was mentioned in my slide. I would encourage people to participate there, and you'll definitely then be able to be aware of what's happening in real time between those two organizations. So I'd say that's where the activity is, and that's where, as a stakeholder, industry stakeholder would point to. I don't know, I'll defer to Ron or TJ if they have anything else to add to that collaboration with Europe on substances.
Thank you for responding to that question.
Sorry, I'm just going to respond to Vada. Yeah, I agree. EMA and FDA have been collaborating on substances for quite a long time with respect to the GSRS system and now the various implementations of the EU SRS system. So there's a very close collaboration with EMA on substance.
Looks like we're just about out of time, but maybe we can squeeze in one more question, and this question will be directed to Dr. Fitzmartin. And here's the question. Is PMDA aligned with the general approach being taken by FDA, EMA, and UMC, or are there key challenges to harmonization based on differences of conceptual approach?
Yes, thank you. That's a very good question. Certainly, and PMDA, Japanese regulatory agency, we certainly cannot answer for PIMDA whether they're aligned with the approach taken by FDA, EMA. But what we can say is that there's an organization, many of you probably on the phone know about the International Pharmaceutical Regulators Forum. That organization has a working group on IDMP. I chair that working group on IDMP for the Regulators Forum. PMDA are members of that working group, participate, attend every meeting. They're very, very interested in IDMP, the IDMP standards, and the progress that we're making. So they're very keen observers, but I think the person who asked that question would have to go to PMDA to direct the question as far as alignment. Thank you.
All right, it looks like according to the computer clock, 10:32 a.m. Eastern Time, that we're all out of time. And so we'd like to thank our panel for the very informative talk and also responding to numerous questions that came in. We do have a few closing reminders. This activity is eligible for an attendance certificate which supports continuing education by SOCR RAPS, SQA, and ACRP. Please refer to our website at fda.gov/CDER Learn for more details. You'll be able to obtain the attendance certificate only upon completion of the survey, which will remain open for 2 weeks. And that's February the 10th is when it closes. Please remember to download the certificate immediately upon completing the survey. It cannot be retrieved after the fact. Also, please let us know your comments and feedback via the survey. This will help guide the design of our future events. On behalf of CDER Small Business and Industry Assistance, we hope you enjoyed today's presentation and found it helpful. We look forward to your presence at future webinars, which will be advertised on our website at fda.gov/CDER SBIA Learn. Thank you.